So the species that we're looking at today is a leaf spot fungus. And what that means is it's a fungus that infects a leaf and grows to form a small, roughly circular patch. And it then rapidly kills all of the tissue in that patch and uses the energy that it gets from the plant to produce its spores. So some leaf spot fungi produce spores on the outside of the leaf, and some of them produce spores inside of the leaf. And this is one that produces spores inside of the leaf in small spherical structures, which are called pycnidia. And those are like a little sphere that's embedded near, in this case, near the top of the leaf, sometimes near the bottom of the leaf. And when they're mature, when the spores are ready to disperse, it forms a small pore in the top of the leaf, and the spores can escape through the pore and be blown around on the wind, and then they can go on to infect more plants. So this leaf spot, you can see it's formed a white sort of rough circle of dead leaf tissue, and then there are small black dots, and those are the pycnidia. And if you look at it under the hand lens, you can actually see there are some very, very small pores in the top of the pycnidia if you look very, very, very closely, because the pores are very small. And those are the things that the spores escape through. So now I'm actually looking at the leaf spot under the microscope itself, which isn't really what the microscope's designed for, but you can shine the light through it and you can still see quite a lot of detail. So I can actually see the pores in the top of the pycnidia quite clearly. And if I go to times 400, I think I should be able to just about see the spores that have escaped. They're sitting around the mouth of the pore, and when the wind comes, they'll just get blown away to infect another plant. The equipment that you need, obviously you need a compound microscope to look at the spores underneath high magnification. You need some slides. You need some cover slips to go over your slides. You need some water in a syringe or an eyedropper. You need a hand lens to be able to look at your sample and see what parts of it you want to look at under the microscope. You need a sharp blade, so you could use one option is to use one of these razor blades that you can buy in a pharmacy, or you can also use these like a scalpel or something like a Stanley knife could also work. The most important thing is just that it's something really, really sharp. You also will find useful to have some tweezers or forceps just to pick stuff up, and a pencil, which we'll use later to squash the sample. So the sample I have to look at is this leaf spot on a garden hebe. So I think this is Veronica franciscana, which is a common shrub that's used for hedges and just as like a decorative plant. And you can see it's got this leaf spot, which is a fungus in the genus Septoria, most likely, but we don't know which one. So we want to look at the spores under the microscope to be able to find out which one it is. So what I'm going to do first is I'm going to look at the sample underneath the hand lens. And what I'm looking for is fruiting structures. So in leaf spots, you want to look for either spores that are produced on the outside of the leaf or small dots, which are pycnidia, which produce spores inside of the leaf. So in this case, it has black pycnidia on the upper side of the leaf that contain the spores, so we want to sample those. So what I'm going to do first is I'm going to take my sharp blade, I'm going to take my sample, and I'm going to cut out a section that I saw under the hand lens had pycnidia. And I'm doing this dry, so there's no water, and that can be quite helpful because it allows me to cut really thin sections. I'm just going to make fine parallel cuts So I basically want to get lots of very thin linear sections of the leaf. And the basic idea is I'm hoping that some of those sections will slice through a pycnidium and release the spores. Yeah. And sometimes when it's dry, it's sort of under tension, so it kind of goes everywhere. So sometimes you do actually want to add water just to stop it from going everywhere. Okay, and then now I'm going to add a few drops of water with my syringe. Water's also sort of gone everywhere, but that's okay. And I'm just going to sort of mix it around. Okay, so the next step is to add a cover slip. I have these nice rectangular ones. And we're going to look at the sections we just made under the microscope. Turn the light on. So what you'll find when you make these sections is you end up often slicing near the pycnidia, but none of the spores actually come out, which can be quite frustrating. So what we've managed to do in making lots of parallel cuts is we've managed to get one of those cuts to cut across a pycnidium, which is this sort of roughly a sphere embedded inside the leaf. And as soon as you cut it open and add the water, all the spores burst out if you make the cut in the right place. But that can be quite hard. Yeah, there we go. And I'm focusing the condenser as I go and adjusting the brightness of the lamp. Both of those things are obviously beyond the scope of this video, but they're very useful for getting your sample in focus. So you've actually got quite a good look at the spores there. Good enough that we'll be able to measure them and work out which species of Septoria this is. So I'm sure it's a Septoria now because it has these long, colourless, rod-shaped spores. In the case that we couldn't find any spores, but we did see that there were pycnidia and just that we hadn't sectioned through any of them well, the technique that you use once you've had a look under the microscope is to take something with a flat base like a pencil and just drop it on top of where your sample is in the slide, and that sudden shock will force out the spores and the contents of the pycnidium. So that can be a really useful technique, although it does get water everywhere. Yeah, if you can't find spores initially. So I'll look at it again and hopefully there will be lots of spores everywhere, and there are. Yeah. And it's also made the slide a bit thinner, which helps to get everything in focus at the same time. So I'm looking at these spores and I can see that they're between about 10 and maybe 30 to 50 micrometers in length. Some of them are aseptate, so they're all a single cell. Some of them are one, and I think even some of them are two septate, which means they have septata, which are divisions that divide them into multiple cells. And on my recording software that's connected to the microscope camera, I can take an image and I can then measure it, and it's all been calibrated so I can measure quite precisely. I think we can quite concretely say that this is Septoria exotica, which is the species that I thought it was going to be, which is the most common of the species of Septoria that you get on garden hebe, and the most common leaf spot on this one in particular, which has 15 to 35 for the length of the spores, which matches our sample pretty much exactly. So I think we can concretely say that that's what it is, which is not at all surprising. So that's quite, yeah, as expected. So that's how you look at leaf spots under the microscope.